Very good morning to all of you. Welcome you all in the ISFCP dialogue series under the Aegis of IQAC-IIC ISFCP, organized by ISF College of Pharmacy and Autonomous College, Moga, Punjab. Today's guest and speaker, Dr. Shivani Chandel, Associate Professor, Department of Pharmacognosy, and its title speech is Principle of Quantitative PCR. We welcome you, ma'am. So you are requested to please proceed further.
Good afternoon, everyone. Myself, Shivani Chandel. Currently, I am working as an associate professor in the Department of Pharmacognosy in ISFCP Moga. So today, my title of talk is Principle of Quantitative PCR. So before going into quantitative PCR, let's first discuss about what is PCR. So PCR stands for polymerase chain reaction. It is a process that identifies and multiplies a specific part of the DNA in a biological sample. So we have a complex DNA sample in which we have a region of interest which we want to amplify, and we can amplify with the help of PCR. So this technique is usually employed in genetic testing, pathogen detection, drug development, crop modification, and forensic analysis. So what happens in PCR is it consists of three major steps. First is denaturation, then annealing and extension. So what happens in denaturation? The double-stranded DNA is denatured at higher temperature, that is 94 degrees Celsius, and it gets converted into single-stranded DNA. And then our region of interest we can amplify that region with the help of specific primers, which we design synthetically. So in the annealing, what happens is we give externally primers, forward primer as well as reverse primer, which binds to the specific part of the DNA. This annealing takes place at the temperature region of 50 to 60 degrees Celsius. When this gets annealed, now the third step is extension step, which occurs at 72 degrees Celsius with the help of tagged DNA polymerase. So what happens? This tagged DNA polymerase with the help of DNTP synthesize new DNA strands, and hence we get multiple copies of our DNA. So this is the basic representation of the technique which I have told. So we take the gene of interest, we set at 94 degrees Celsius, then we anneal at 50 to 60 degrees Celsius depending on the primer GC content, and we extend at 72 degrees Celsius. So usually the number of cycles which we employ in this PCR technique is 25 to 30 cycles. So what goes in conventional PCR? We have a template DNA, pair of DNA primers, DNA polymerase, DNTPs, and a buffer which help in maintaining the pH and provide magnesium. So what does this magnesium do? This magnesium helps in the binding of DNTP hydroxy part. It helps in making phosphodiester bond between primers as well as this DNTP. Sorry. So in this reaction, what we can see from the graph, we have an exponential phase as well as a plateau phase. So in the exponential phase, what happens is with the number of cycles, our PCR products get amplified, and at plateau phase, this gets saturated. So early in the reaction, in the exponential phase, as you can see, there will be very few copies of the target sequence, and the amount of PCR product will grow slowly. Now in the plateau phase, what happens is, in the exponential phase, the amount of PCR products will grow faster and faster until there is no enough reagents to sustain the reaction. So PCR, as we can see, it is an endpoint assay. Now I will explain it why. So traditionally, PCR simply aims to obtain a large amount of target sequence. So researchers are trying to get the PCR close to or the plateau phase before stopping the reaction. So typically the exponential phase of the reaction is, we can only monitor the plateau phase. We can only see whether there is amplification of the gene or not, but we can't really quantify it exactly. So endpoint is the only point where we can see, okay, our PCR has completed. So we have different starting points, but we have the same endpoint. Like we can't really identify at what cycle our PCR product has completely, or our PCR reagent has completely exhausted. So for this reason, we have an advantage in qPCR, as it can quantify at which cycle uh, the reaction has stopped. So qPCR changes the approach to by gathering information during the reaction when different reactions are still in the exponential phase. So qPCR allows us to monitor PCR products during exponential phase, as it's doubling with every PCR cycle. So what really goes in the PCR? So qPCR are basically the same as regular PCR with the addition of a fluorescent dye or a probe. So what we add in this reaction is just one additional thing we are going to add here. DNA is there, primer is there, DNTPs are there. Just only what we are going to add extra is a fluorescent marker. So what this fluorescent marker or fluorescent probe will do, it will intercalate between the our target gene or target DNA. So if there is any amplification, so the fluorescence signals will tend to increase. qPCR fluorescent dye will be there. So as I have previously told, so qPCR uses a fluorescent dye that only fluoresces when bound to double-stranded DNA. So every PCR cycle, more and more double-stranded DNA is made, and fluorescence will get brighter and brighter. So usually what we employ in this qPCR is cyber green one. I've given the structure as well as the name in this slide. So as you can see, when there will be this fluorescent probe, so it will enhance the fluorescence. So the amount of fluorescence in this reaction will serve as a readout of how many copies of DNA have been made. So as you can see in the graph, we actually calculate with the help of relative fluorescence and number of cycles. So how it will be detected, I'll be again explaining in further slides. So early in this reaction, there is a little DNA that then fluorescence can't be detected, as you can see from 0 to 20 cycle. But eventually there is enough DNA that can fluorescence, and then it can pass the threshold level, which is the minimum level to pass where it can be observed. Okay. So the cycle at which the fluorescence is first observed above background is called CT for cycle threshold. So as you can see, for different kind of sample, I have labeled in multiple colors: green, gray, blue, and orange. So a reaction CT is directly linked to the starting concentration of a target sequence. In a PCR, the amount of target sequence double every cycle. This means that if a reaction reaches CT one cycle early than other reaction, it is expected to have started with twice as much as target sequence. So with the help of example, if you can see, this green one is going exponentially before than the other gray color. So if there is a 2 cycle difference, as you can see, the number of cycles in between 15 and 20, there are some markings are there, like 1, 2, 3, 4. So every marking denotes one cycle. So 15, 16, at 16 green is going up, and then at 18, the gray the molecule with gray representation, or the tube with gray representation sample, is going up. So if there is a 2 cycle difference, you can expect a 4-fold difference in the starting DNA concentration. And if there is 3, so with number of cycle, it goes 2 raised to power n. Okay. So as I've previously told you, these two reactions reach CT 2 cycles apart. So the reaction representing green line is expected to have started with 4 times as much as of the target DNA sequence. So if we compare these gray and blue ones, so as you can see, the gray one has started at cycle number 18, and the blue one has started as cycle number 22. So the reaction represented by the blue line, which is CT4 cycle after the gray one, so which reaction we can expect is around 2 raised to power 4. So the DNA change, DNA difference between these two samples is around 4 times. So qPCR also allows researcher to estimate DNA concentration, as we have previously seen with the help of example. So by comparing PCR reaction in real time, qPCR allows you to calculate the relative amount of starting sample between two reactions. Now imagine if you want to detect virus load in the sample, which was you must have heard this technique in COVID-19 when the testing was done with the help of qPCR. So what happened in this technique was the basic principle which we have previously discussed. So we purified the RNA from the sample or from the swab which we have collected from the patient, and then what we did was we transcribed into cDNA, and then with the help of qPCR, which and the cDNA acted as the template. So with the help of qPCR, we amplified and detected. So if you can see in the graph, result part, signal to cycle, the green line is showing positive and the gray line is showing negative. Why? Because if you have higher DNA, I mean the higher titer of the virus load, then the amplification will takes much faster. And if the titer is less, then amplification will take at a further till the end. So if the titer is less, that means this give negative result, and if the titer is high, then it will start amplifying much higher, much before in the number of cycles. So with the help of this, we can identify whether our person or the sample is infected with COVID-19. So I hope whatever I have delivered, you are able to catch that, and if there are any further questions, you may ask. And thank you for the PPT.